Ladies and gentlemen, I would like to welcome you to the June 2023 Pacific Coast Archaeological Society meeting and presentation. Thank you all for coming. I just want to ask if anyone has any announcements, either directly with the Archaeological Society, or if you're aware of any programs or meetings, events coming up that you think would be of interest to the listeners here.
I would like to request that you approach the microphone when you speak. Otherwise, it's barely audible. Thank you.
Okay, I'll bring it a little closer for the time being. Megan, there was some discussion prior to the meeting of the grants that had been awarded. Did you want to make an announcement about those grants and who they were?
I don't have the information in front of me, Steve. I'm kind of behind the loop right now. I spent last night in the hospital, so I wasn't really prepared for this meeting.
Well, in that case.
We awarded 3 scholarships, and it appears I have all the paperwork, and those checks will go out likely in August is when we usually send them their awards.
All that information is in our June newsletter, which is online.
Reen, yourself, I know that we have an issue of the PCS Quarterly coming out shortly. Do you have an idea of when that might be printed? And
the interior part of the volume 56, number 2, has been printed. I received a call today that although they thought they had the inventory, they do not have the inventory on the cover, and they don't expect to have it. So we have a problem. I don't want to use a pink cover. I don't think that will go with the goldenrod. So I think we have to probably change to a different cover, but it will not be pink. I will be in touch on that.
Okay. All right. So thank you for that. As I said, any other announcements or people know of events coming up that would be of general interest? Then with that, I will turn this over to Joe to introduce our speaker this evening.
Stephen, can you make one more announcement about going mute?
Yes, please. Look on the screen here. There's still a couple people that are live.
Okay. Thank you very much, Steve.
There we go. Thank you all.
Good evening, everybody. If you're a first timer, we welcome you. If you're more than a first timer, we still welcome you. Welcome, everybody. This is our last meeting of our program year. We'll start up again in September. I'll make an announcement of the September speaker after Nathan's talk. Tonight we have Nathan Nakatsuka, and he is coming to us from Hawaii. Hawaii. And the title of his talk is Genetic Evidence for Ancient Population Shifts and Migrations in Central and Southern California. Nathan was born and raised in Honolulu, Hawaii. He attended Harvard College, earning an AB in chemical and physical biology. He then attended an MPhil in genetics from the University of Cambridge on a Gates Cambridge scholarship, working at the Wellcome Trust Sanger Institute. Nathan received his MD at Harvard Medical School in the Harvard MIT Health Sciences and Technology MD track, and his PhD from the Systems Synthetic and Quantitative Biology PhD program, working in the laboratory of David Reich on population genetics of South Asia and the Americas. During his PhD study, the majority of his work focused on studying ancient DNA to uncover the ancient history of North, Central, and South America. He currently has a postdoctoral fellow, or he is a postdoctoral fellow at the New York Genome Center in the laboratory of Raul Satya, working on single cell analysis and neuropsychiatric traits. I have the high honor of introducing and welcoming Nathan Nakatsuka. It's all yours, Nathan.
Okay. Can you see my screen? Is that working?
Yes.
Okay, great. Yeah. Hi, everyone. Thanks, Joe. So I'm Nathan. I'm both a postdoctoral fellow right now with Rahul at New York Genome Center, and I'm also working on this project as well as a postdoctoral fellow with David Wright, kind of following up on some of the PhD work I did during my PhD. So this project was done in collaboration with John Johnson at Santa Barbara Museum and Paul Langenwater at Biola University. So before I start my presentation, I really want to thank the organizers of this conference, the PCAST event, and just being able to talk to all of you. I also want to acknowledge the indigenous people on which the PCAST event is being held, the Tongva in particular, as well as the Chumash tribes to the north, and the Occhichemen, or also known as Juaneño, and the Payom-Kuwechem, or Luiseño tribes to the south, some of which I'll be talking about today. And so today I'm going to be talking about an ancient DNA project on California and Northwest Mexico. So I'm going to speak primarily from a genetics perspective, but I'll also integrate in archaeology and linguistics evidence. I'm not an archaeologist or linguist, and because many of you are, please correct me if I say anything wrong. You can use the chat feature to put in any comments or questions that you have throughout the presentation. I'll try to stop if I can see some of the questions. If I don't get to them, then I'll try to answer them at the end. So first I'll start with this presentation with some background on ancient California that most of you probably know, but some of you might not know. So this is kind of from a genetic and archaeologist perspective. I want to talk about a bit about the region we now call California as one of the most unique and important places for Native American history. So the history of the people in the Americas began at least 16,000 years ago. It was thought that one of the routes by which the Native Americans traveled was along the West Coast, with some of the people migrating near the ocean. And another group might have come down several thousand years later through the middle of the continent when some of the ice sheets separated. But California, in particular, Channel Islands, are a very key region for the peopling of North America, because there appear to be people who stopped there along their journey and lived there continuously for at least 13,000 years based on human skeletal material, stone tools, and other human artifacts. The extremely early peopling of California is evidenced by the fact that there's great diversity of Native American languages in the region. So if you think about Europe, where that region, almost everyone speaks Indo-European language, this is very different from California, where there are very highly divergent language families that are in California with split times over 10,000 years ago. For example, in Northern California, we have some languages like the Algonquin and Athabascan speakers. In Central California, that's the region where San Francisco and Palo Alto are the Yushian languages, such as Ohlone. And in addition to the Yushian languages, there's also a very ancient language family called the Hokan language family. That's kind of a hypothesized language family that includes such languages as Esalen. There's also another large language family in Southern California called Uto-Aztecan. There's another possible language family called Yukian, as well as some other, the other Uto-Aztecan languages are languages such as Tongva and Luiseño, which I'll talk a bit about during this presentation as well. So here's a broader map of the Uto-Aztecan languages. This is really important for a really important archaeological question and kind of linguistic question about where did the Uto-Aztecan languages come from? So one hypothesis is that they came from central Mexico with farming, and then there's kind of a great expansion of people when farming happened, and people were able to grow to large population sizes, and this population expansion led people to move out to various places, both south as well as north. So Peter Bellwood's an archaeologist, and Jane Hill's a linguist, the late Jane Hill was a linguist who were major proponents of this hypothesis. Another hypothesis is that it originated in the Sonora Desert from hunter-gatherers between 3,000 and 5,000 years ago. I think Lyle Campbell in the past has proposed this view. He's a linguist, and they suggest that it was caused by climate change, with some going south to start farming and others going up north. Some others, like David Shaw, believe that the proto-Uto-Aztecans were from Central California Valley. And others, such as William Merrill and many others, propose the Great Basin as the origin. And some others have proposed Central Arizona as a source population. So a lot of these are based on linguistic arguments. There's some archaeological arguments as well, but the linguistic arguments are based on loan words and different cognate roots. And basically what they're trying to do is construct an agricultural vocabulary that encompasses all of the languages and see the different split times and when they might have arisen. So I'm not an archaeologist, so it's difficult for me to assess these arguments. But one thing that some of the linguists have agreed on is that it seems that maize came to the language family after the diversification of language family. So that might have be something that's relevant to this as well. So a bit about California oral indigenous history. So there's a lot of different California indigenous groups that have a lot of different oral histories that are really important for informing how people were created and how humanity relates to nature in their worldviews. So Chumash oral history relates to, has a lot of different kind of stories about the relationship of how the people were related to different animals as well as different creators. So in one story, there is a rainbow bridge that was created by Mother Earth, Hutash, and they kind of people were sprouted from Santa Cruz Island, and as the population increased, Hutash had to create a land bridge that allowed cross-island travel and travel between the islands and the mainland. They also have an alternative creation story about this coyote actually creating people from a marble tablet. The Ohlone have oral narratives about creation after flood by either a coyote or a feather from an eagle. And Tongva have creation story about a first man and a first woman who had guidance from Kuaar, who received the soul of all who died. And basically all of these oral histories usually show a strong connection of people to their homelands and also to the nature around them, but also with resilience to change. So we did a lot of discussions with indigenous groups and archaeologists, and there are several key questions we wanted to address with this study, including if, first, if there are interrelationships between within California as well as between California and other regions. We wanted to know how the people on the Channel Islands related to those people on the mainland, and also how much genetic continuity is there amongst the different ancient individuals over time, and if there's continuity between the ancient people as well as the modern people who exist today. Then a bit about the language families. How do the different language families arrive in the region, and what are the original source populations of the language families? And then there are additional questions about, so an additional question is also about population size, but there are also additional questions that will be answered in future studies that will not be addressed here, such as more recent population movements that relate to the more recent history of the Chumash and Ohlone and other groups, and also how did the ancient people relate to people living, who are literally living today. So I'll give some background now about genetics and ancient DNA and kind of why we use ancient DNA, how it interacts with archaeology and linguistics, and how the different pictures of history can all be melded together. So this is a very basic background about basic genetics. So humans, as we all know, have 23 pairs of chromosomes, and 23 of them come from your biological mother and 23 of them come from your father. So you pass down one pair of these chromosomes through each generation, but very importantly, there are random changes such as mutations and recombinations that happen over time. And this leads to changes that we can track and measure. So people who are more closely related to each other have fewer genetic differences between each other. And the same goes for larger populations where groups that are more closely related to each other have more similar genetic sequences. And because these patterns of DNA are passed down are very predictable, we can examine the DNA of individuals and see how they relate to each other on an individual level. That's kind of what you do in forensics. But you can also look in a larger population to see how they relate to each other and what populations specific individuals are from. So ancient DNA is very important in this process. So you can sequence people who are living today, but also people who lived in the past. And ancient DNA is from humans or other organisms that lived many years ago, often thousands of years ago. This is obtained from skeletal material of the ancient humans, and it's usually brought into a clean room, and a small amount of the bone is then powdered, and DNA is purified from this material and then sequenced. So you might ask, what's the advantage of ancient DNA as opposed to DNA from present-day humans? The big advantage of ancient DNA is that we can now observe genetic variation, not only in geographic space, but also over time. So you can compare the genetic profiles of individuals from the same region over time to see how the genetics of an area is changing over time, such as new people moving into a region. One thing we can use to track human history is mitochondrial and Y chromosomal DNA. So mitochondrial DNA is DNA that's passed down exclusively from the mother. So it shows history from human history from the maternal line. And then you can look at Y chromosomes as well that are found only in males, because the sex chromosomes of males are XY, whereas genetic females are XX. And analyses of Y chromosomes show human history from the paternal line. So here's kind of a figure of this from a mitochondrial perspective. The mitochondrial DNA is passed down from different females down on the maternal lineage, whereas the Y chromosome is passed down just from the paternal line. So mitochondrial and Y chromosome DNA can reveal some aspects of human history, but the autosomes, which are the non-sex chromosomes, are usually more powerful for revealing mixture between populations. So I often use this example when we're teaching Native American high school students, some of whom might be part Navajo, part Hopi. So if you're a child with a Navajo mother and say a Hopi father, you have one set of Navajo chromosomes and the other set of Hopi chromosomes. And so if it keeps happening over generations, each generation will have smaller and smaller fragments of these and that are mixed up more and more. And so you can use statistical analyses to infer these events and the fact that mixture happened and also when it happened as well. So yeah, if there's any questions, I guess there's no questions. So yeah, there's important quality control steps for this process. One of the most important ones is reducing contamination. So we need to ensure that DNA is human and not bacterial, and also that it's ancient and not from modern humans. So first, to ensure it's human, we can map the DNA sequences to the human genome. And then you can also check that the DNA is ancient, meaning thousands of years old, and you're looking there for DNA with characteristic damage. There's also multiple other ways to determine if there's human contamination. So we can look for alternative ancestry, such as say European ancestry in ancient Native Americans. And for male ancient individuals, we can look at heterozygosity, which is two different types of variants on the X chromosome, since genetic male should only have one X chromosome and therefore no heterozygosity. And lastly, there's this other method that we developed called ContamLD, which works for both females and males. And it's based on the breakdown of linkage disequilibrium on autosomes. It's a very technical sort of thing that you could ask me if you're interested in genetics a lot. So when you're making historical inferences with ancient DNA, we need data of sufficient depth and quality. This usually means at least 200,000 SNPs, which are genetic variants. And it's also helpful to have many individuals, but even one individual can tell you something, such as whether a particular ancestry type is present. More individuals usually help you to tell, such as if there's genetic heterogeneity in a region. And so one example I often bring up is, say, an alien comes in from 20,000 years in the future, and they're looking at DNA of people living in New York City today. And so if they get a single individual's skeletal material and DNA from that individual, they might say, oh, there's someone who is Italian ancestry. And if they keep sequencing more and more people throughout New York City, they'll see, oh, there's Peruvian people, there's Chinese people, there's Nigerian people, et cetera. And as you sequence more and more people, you'll see the genetic heterogeneity there, but a single person will at least tell you that there's that type of ancestry in that region. So now we can get to the results about California's history in particular. The only human whole genome studies previously published in ancient California were in 2018 and 2022. The 2018 paper focused on broader global Native American ancestry components rather than the more recent history. while the 2022 study found evidence for genetic continuity in Central California over the last 2,000 years. So in this study, we use the data generated from those studies, as well as sequencing 81 additional ancient individuals who lived in both Southern and Central California. And, oh, sorry, I'll talk a bit slower. And 40 new ancient individuals from Mexico, primarily from the Northwest region. So these individuals are existed about 8,000 years ago in California, as well as about 2,500 years ago in Mexico. So prior to the study, we consulted with the indigenous groups in the regions near where the ancient individuals were found, including several Chumash tribes and Tongva in Southern California, and Ohlone and Esselen communities in Central California, as well as the Mexican government and indigenous Mexican communities in Sonora and Northwest Mexico. We also held several meetings throughout the study to return results as they came in to ensure that the study was conducted and the results were reported in a way that centered indigenous perspectives and the questions they were interested in. So here's a map of where the ancient individuals in the study lived, encompassing Central California in purple, Southern California mainland in red, the Northern and Southern Channel Islands that you can see there as well, and Baja California in light blue and Mexico in blue. Here's a map of the Channel Islands. The ancient individuals in the study are from the 6 main Channel Islands. The Northern Channel Islands are distinct from the Southern Channel Islands because inhabitants of the Northern Channel Islands, when contacted by Europeans, were found to speak Chumashan languages, while the inhabitants of the Southern Channel Islands spoke Uto-Aztecan languages. Now I'll get into the results. So one of the things you can do genetically is to produce these trees of people to see how they relate to different people. In this case, we're using this statistic called the outgroup F3 statistic, which shows genetic similarities between the different groups. And as you can see here, the groups generally cluster by geographic region. I'm coloring them by region, but it's not fully by geographic region. So there's an interesting cluster in the middle, this section that's in black, that involves the oldest individuals from Northern and Southern Channel Islands and then the Southern California mainland. And as you can see, the reason there's not fully geographic clustering is that there's a certain Mexico ancestry that's coming in. And these individuals there in the black section are individuals who lack this Mexico-related ancestry. And later on, you see people in the late Northern Channel Islands and other places that are clustering different because they're clustering more by age than by geographic region, because they have more of this ancestry that's Mexico-related, which I'll talk about soon. Here's another way to cluster the data. It's called an MDS plot. It also shows mostly clustering by geography. And the reason it's clustering by geography is that people who are living in the same region tend to be more genetically related to people in those regions, even over time. But the fact that there's not a full genetic clustering by geography is that the fact that there's changes in ancestry over time. So the mitochondrial results is the first thing we looked at, and these were very striking. When we looked at individuals over 3,500 years old, 30 of the 36 have the A2 haplotype, and the only ones without the A2 haplotype were all from mainland Southern California. In contrast, after 3,500 years ago, only 35 of 91 have the A2 haplotype, and many other haplotypes are represented after that time. And this all points to possible migration into the region from outside. So if you look in the Mexico individuals, they have a lot of these extra haplotypes that likely were the haplotypes that are brought in from Mexico into these regions. You can also look at Y chromosome results, and almost all of them were this one Y chromosome haplotype Q1B1A, and this is the most common haplogroup in Native Americans today. This is different than in very ancient South Americans, where the very ancient South Americans have a lot of a different haplotype. So that points to a different history that is present there in the past. So we next wanted to use more rigorous analyses to determine if any ancestry came into California from outside. So to do this, we compared the genetic profiles of the oldest Santa Rosa individuals to the younger Santa Rosa individuals, and also other California regions. So we used this statistic called the F4 statistic to assess whether the younger Santa Rosa individuals had evidence of mixture from other groups. And we found that the only groups outside of California with evidence of having mixed into California are a few Northwest Mexico groups. This is very interesting in that these groups are all groups that likely spoke Uto-Aztecan languages, and they're very similar ancestry to present-day people in the area, the Odom or the Pima, who speak the Uto-Aztecan language. So here's a graphic of this. People coming in from Mexico over time, Mexico-related ancestry at least over time, is coming into California. So on the y-axis is the amount of Northwest Mexico-related ancestry, and the x-axis is the age of the individuals, with the left side being older and the right side being younger. We find that most of the earliest groups in Southern California have no evidence of this Mexico-related ancestry, including the earliest individuals from the Southern Channel Islands and the Southern California mainland. And this ancestry slowly increases over time around 5,000 years ago and reaches a very high level in the Southern Channel Islands. And this is consistent with the fact that the Southern Channel Islands were known to have spoken a Uto-Aztecan language when the first Europeans met them, such as Nicoleño, or now even the Tongva speak a Uto-Aztecan language. The Northern Channel Islands, in contrast, spoke Chumashan languages, which are not Uto-Aztecan. So basically, people with ancestry related to Uto-Aztecan speakers are coming into from Mexico, or at least Mexican-related ancestries coming into California and leading to a potentially language change in the Southern California, Southern Channel Islands, but not in the Northern Channel Islands. So we also wanted to tell if the gene flow was unidirectional or bidirectional. So we compared the oldest Mexico individual, who was 2,500 years old, to the younger individuals who were 1,100 years old. And the younger Mexico individuals had no California ancestry relative to the older ones. So it shows that there's unidirectional mixture that's coming in just from Mexico into California and not from California back into Mexico. So I look a bit washed out because there's some sun coming in. But anyway, this is also potentially a small bit of evidence against a Central California or Great Basin origin of Uto-Aztecan, because it points away from substantial migration from these regions between 2,500 and 1,100 years ago, which would be likely if there was a migration of Uto-Aztecan speakers down to Mexico that occurred prior to that period. We also note that one of the proponents of the Great Basin hypothesis, William Merrill, made pretty strong claims about the fact that there was mitochondrial studies that show that there's no genetic evidence for migration from Mexico to the American Southwest. But here we are providing evidence that there is migration from of Mexico-related ancestry into California. And so this would cause some people in these with these hypotheses about a Great Basin origin to revise their arguments there. Okay, my face is getting washed out. Okay. As noted before, there's some linguistic evidence and archaeological evidence for Mexico influence in California, such as the contracting stem dart point. However, our results don't actually rule out any of the previously suggested hypotheses for Uto-Aztecan origins, but they do require that some of them be revised. For example, maize agriculture was only present in the American Southwest after, sorry, I'm gonna move the screen. Maize agriculture was only present in the American Southwest after 4,000 years ago, which was after our inferred date of the Uto-Aztecan related migration into California. So we have evidence now that there's migration from at least 5,000 years ago and from Mexico-related ancestry into California. And this is an argument against agriculture being the only plausible candidate for the spread of large number of Proto-Uto-Aztecan speakers, since clearly we're seeing that there's many people moving into California prior to agricultural spread. Our results don't rule out a hypothesis that it still came from central Mexico, but at least it's saying that there was an earlier migration of people. And if you have that hypothesis, you would have to say that there was this earlier migration of hunter-gatherers and followed by a later migration of farmers. So that's a revision of the prior way that they framed it. Another thing is that the Mexico related in California doesn't necessarily mean that the ancestry came directly from Mexico. It might be Mexico related ancestry that came up to, say, the Great Basin or other place and then came into California. But as I said before, a hypothesis of the Uto-Aztecan community coming from Central California into Mexico now has to account for the fact that there is evidence against a migration into Mexico from California and the Great Basin between 2500 and 1100 BP. So if you're going to use that hypothesis, you have to say that there was a migration that happened earlier and didn't and stopped, basically, which is potentially slightly less plausible. So now we'll look at the purple line, which is Central California. And this is also very interesting because there's no increase in Uto-Aztecan ancestry in this region versus all the other regions you're seeing an increase in Uto-Aztecan related ancestry. And this is consistent. The fact that there is Uto-Aztecan ancestry in the first place in a 5,000-year-old individual in Central California is consistent with archaeological evidence or hypotheses of what's called Old Uto-Aztecan, which is based on linguistic evidence that there were Uto-Aztecan speakers in San Joaquin Valley before 5,000 years ago that were then displaced by Yokutzen speakers. So people who had that hypothesis, now we have some genetic evidence that this is consistent with a Uto-Aztecan speaker being Uto-Aztecan speakers being in that region beforehand, very early on. So now I'll follow that up with some talk about population changes that happen in Central California. As a reminder, here is the area of the ancient individuals from Central California in our study. Most of them are from the coast, but we also have individuals from the inland in Calaveras County. So that's all these purple individuals up here. So for the assessment of the ancestry in Central California, we're doing the same sort of strategy of comparing the oldest Central California individual relative to the younger ones. So when we compared the oldest individual we have, who is from Pacific Grove, to the later Central California individuals, we found a really fascinating signal. The oldest Pacific Grove individual has a very interesting genetic affinity to some of the Paracues groups in Baja, Mexico. These are very dark red dots here. The fact that these are not as dark red might be because they're very low coverage and they're only a single individual, whereas these dots are multiple individuals of much higher coverage. So the Paracues were thought to have spoken a Hokan language. They're kind of isolated groups, relatively isolated groups in Baja, Mexico. And the Hokan language is a hypothesis language family that was thought to be potentially the original language family in Central California and Baja California. And in Central California, it's thought to be the original language there before the Yushin or Ohlone languages were introduced about 4,000 years ago. So the genetic attraction that we see between the oldest Central California individual and the Paracues individuals, the fact that it's not found in the later Central California individuals is potentially evidence of the fact that there is that this was kind of Hokan ancestry that was there a very long time ago, and it's consistent with a new migration into the region that kind of changes the ancestry later on. However, we do see still some genetic continuity between the oldest individual and the younger individuals. It's just that we also find evidence for an additional migration that happened up here in the Central California region. So you might be asking, what was the source population for this migration into the Central California coast? And we don't know yet, but we do have, since we have these inland individuals from Calaveras County, they're a possible source population. We find some genetic evidence for a mixture from there. The issue is that these individuals were also thought to have spoken a Hokan language. So this doesn't neatly fit the Yushin migration hypothesis. So we need more sampling from this area to further test the hypothesis about who's coming into this region, what are the different ancestry types, and how they're changing over time. So the Baja-related ancestry in Central California, as I said, is consistent with an ancient Hokan ancestry that's widespread in Central California. Some of the groups with these languages are the Esalen or Paracues. And there was a hypothesized language change to Yuxian languages after 4,000 years ago, such as Ohlone. And we do find evidence of a migration from inland into the coast between these times. But the fact that, as I said before, the fact that these Calaveras County individuals were thought to have spoken Hokan languages doesn't neatly fit into a hypothesis of Yuxian speakers coming to the coast. And as I said before, even though we see evidence of migration into the region, we don't see evidence for a full displacement. There is still substantial genetic continuity that is still in this region. So one thing that I also wanted to address with this study was whether or not there's a genetic connection between Polynesia and California. So some people have previously suggested there is ancient connections between Polynesia and California that led to the use of the Tomos in the Chumash and Tongva. And some suggest that there is some linguistic connections between these two. The Tomos are this kind of kayak-like canoe structures that allow people to move between the islands and the mainland. And some suggested that they got these canoes from Polynesians. Yeah, that's a good point about the Paracue and Hokan language. It's kind of a hypothesized sort of thing because they don't, the Hokan language as a whole is kind of a hypothesized sort of thing because the Paracue, we don't really have any new Paracues individuals. So, or any individuals who kind of speak the current language because the people group died out many years ago. So this is all, anytime I mention Hokan in the first place, it's all fairly tenuous ground because of Hokan languages are pretty tenuous in the first place. But yeah, so in terms of this Polynesian connection, we don't find any genetic evidence for Polynesian ancestry in any of the California individuals. It's possible that there was cultural connection that didn't involve mating between the groups, but either way, we at least don't see a large Polynesian inflow of ancestry into California. So now I'll talk about some population sizes of ancient California. There are several different genetic ways to assess population size, but two of the most commonly used ways are to use runs of homozygosity or conditional heterozygosity. Those are two opposite things. So runs of homozygosity are kind of things that you see in your genome when your parents are very closely related to each other. So at the very extreme end might be someone who's, say their parents are both first cousins, then you have a lot of regions where both of the chromosomes are very similar to each other. And so you have a lot of these regions where they're called homozygous regions, that they're very similar to each other. And so if you have individuals with a lot of these runs of homozygosity, you can infer that's a very small population size. And the opposite of this is conditional heterozygosity, where this value is smaller when the populations are smaller and larger when the populations are larger. So these are two opposite things that both tell you about the population size. So here are analyses of runs of homozygosity. In this plot, higher bars indicate more ROH regions, which indicate smaller population size. So as you can see here, the northern and southern Channel Islands have more ROH regions than the mainland, indicating they have a smaller population size than the mainland. It also is interesting that the population size appears to increase over time, but evidenced by decreasing ROH in each region. So we can calculate their population sizes through maximum likelihood estimates. And shown here are some of the estimates for the populations based on the younger individuals of each of the regions. And you can see kind of how they're different between the different regions. So when we compare the population sizes of California to other regions, the Channel Islands were more similar to Guam, Saipan, Patagonia, and pre-agricultural Caribbean. A lot of these are islands that are it's hard to have a very large population in any of them. The California mainland was slightly lower than the size of post-agricultural Caribbean, and Mexico was more similar to Peru's population sizes. So, so the Channel Islands have a very small population size because it's very small islands, whereas the mainland has larger population sizes, but still not as large as Mexico, where those population sizes are more similar to Peru, that has a lot of agriculture and a lot of base food availability for having very large populations. So we can look at conditional heterozygosity and see that they're reversed from the ROH estimates, as you might expect, because they're inversely correlated. And so it shows the same patterns for relative population sizes as the ROH analyses. One interesting thing we found is that if you look at the different Mexico groups, the more southern groups have larger inferred population sizes than the more northern groups. We don't currently have a good interpretation for this, but you can speculate that potentially agricultural conditions are better more south, and that would allow larger population sizes, whereas they might be more hunter-gatherer, like in the northern parts, and have smaller population sizes there. So lastly, I'll talk about the relationship of California and Mexico to the earliest sequence Native Americans. So in a prior paper we had previously published, we found that the earliest individuals from California, Nevada, and most of the earliest South Americans have ancestry related to the earliest sequence North American, which is associated with the Clovis culture, which is a hunting type of projectile point. This ancestry was mostly displaced in South America, but we find that the ancestry was preserved in California up to the youngest individuals. And then we find, most interestingly, that the Mexican individuals have no evidence of this ancestry and instead have a more basal ancestry that must have come from a different migration. Oh, good question about NE is effective population size. So I'll go back to the prior slide. Wait. So yeah, here NE means effective population size. Effective population size is basically what is the population size if there was equivalent to freely mixing people. So if there was a free structure where everyone randomly mixed between everyone, then that would be the approximate population size. So that effective population size is always much less than the actual population size, because the actual population size is usually a bit structured, because people tend to marry people who are, or have kids with people who are, might be from the same region as them, or some same, ancestry type or something like that. And so these are usually maybe 3 to 10-fold lower than what the actual population size is. Oh, it's good. Elaborate on the ancient connection of the maritime Pacific coast travelers from the northern Ecuadorian coast going way back from the exchange of and maize domestication with the southern. Oh, this is a very interesting question. So you don't see that they ever traveled further north and mingled with the native populations of the Channel Islands in Southern California. That's an interesting question. So, so the question here is about whether there was, kind of a network of people coming down the Pacific coast and going back up the Pacific coast. So basically, you can have an exchange of different things. On the coast, you sometimes might have seaweed or seafood, and maize domestication. Basically, do you see that there do you see that there is ever travel further north from the kind of more southern populations all the way up? So the only thing that we saw was basically people coming from Northwest Mexico. When we looked at people from Central Mexico or Southern Mexico, they didn't have evidence for this mixture up north. It's possible that there was some mixture that came up, but currently we don't have good evidence that people are coming, say, all the way from Ecuador or further north. say Belize or like then we have Nahua individuals and their ancestry don't appear to be the thing coming up to California. So I hope that answers your question a bit, but yeah. But yeah, back to this point about the earliest sequence people, we have evidence that this Clovis culture individual Clovis culture's ancestry comes down to these different regions, but is it different from the ancestry that comes into Mexico? Oh, good question about what sites are the Baja California samples from. So let's see, go back to this. So the Baja California samples are not particularly old. We really want older individuals from there. We want these, I think the oldest one is 500 to 1,000 years old. So we can get much better information if we start having individuals from Baja Mexico or Baja California that are much older. So if we can start finding individuals from there who are 5,000 or 10,000 years old, or even 4,000 to 6,000 years old, that would help us a lot. But currently we're making a lot of hypotheses based on these individuals who are relatively young. So all of our hypotheses are slightly fraught by the fact that there might have been additional mixture that comes into these individuals, and they might not reflect, the ancestry that was in Baja Mexico or Baja California, say 5,000, 6,000 years old. But yeah, we definitely want to find Baja California individuals who are older than the individuals that we have. Yeah, so getting back to this. So this is kind of the end of my presentation. So kind of summarizing it, we have a finding that ancestry related to Northwest Mexico Uto-Aztecan speakers came to California at least 5,000 years ago and increased in proportions over time in most of the different regions. And they reached their highest levels in the Southern Channel Islands, where Uto-Aztecan languages were spoken at least at the time of European contact. This is important because it impacts current models of origins of the Uto-Aztecan language family, and it kind of will cause people to have to refine their hypotheses or their models of how the Uto-Aztecan language family arose. We find that population migrations occurred into the Central California coast, but there's still genetic continuity over time there. This is consistent in some senses with the arrival of the Yuxian languages into the region, but not perfectly consistent because of, as I said, the individuals that we have were not Yuxian speakers. We don't find evidence of Polynesian ancestry found in ancient California over time. And we find that the population sizes of the Channel Islands were smaller than the mainland and similar to Patagonia and pre-agricultural Caribbean islands, but increased over time. And lastly, we find that ancient Mexicans harbor a distinct ancestry from that found in ancient California and South America. So I'd like to thank the Chumash, Tongva, Ohlone, and Esselen and Mexican communities as a whole for supporting and assisting this work. In particular, Manuel Armenta, Rex St. Onge, and Robert Dorme, as well as the late Ella Rodriguez, who helped substantially with the study. Brian Hogan is also from the Chumash community, and he helped a large amount in the study. He's a PhD student in archaeology and anthropology. And I would also like to thank the members of the Reich Lab, John Johnson, and Paul Langenwater as well as Jose Luis Punzo and his colleagues in Mexico. Thank you for listening to this presentation. I can take any questions now. Yeah, people can either chat questions or ask questions in person.
I'm not sure how to pose this question, but the one question was going to be from influence, Polynesian influence, and you answered that one. But is it possible that there could be a European influence that came into the Mexico or the Central America and then went north?
Yeah, that's interesting. We don't have, I mean, I guess by the time Europeans came in, that we're not looking at that modern of individuals usually. So it's possible that that happened, but we wouldn't see it very much because we're looking at people, say, older than 500 years ago. So if there was that, obviously that happens now in like, 100 years ago, 50 years ago, even present, but we aren't really addressing that because we're looking at people older than 500 years ago. Good question about which modern U.S. indigenous tribes speak Uto-Aztecan derived languages. So I have lots of friends who speak Uto-Aztecan languages actually. So Luiseño is a tribe in San Diego that speaks Uto-Aztecan languages. The Tongva are another tribe. They're also called Gabrielino. So they're in LA that they speak Uto-Aztecan languages. Yaqui in Arizona and Mexico are another group. Tarahumara, another group. Actually, I have friends in all these groups. Hopi is a Uto-Aztecan language as well. And Shoshone is one that's pretty far up in close to Idaho even. And Nahua are definitely one. Actually, that's not modern US though, but Nahua in Mexico. Yeah, so those would be some of the major ones that are speaking these languages. So yeah, many of these are languages that are doing reasonably well in the sense that unlike say Esalen or some of these other languages, I mean, for example, Luiseño, they have things like video games to help people learn Luiseño languages. And there are people who learn Yaqui in different ways. Some of these other languages are reasonably vibrant that are have a good chance of being continually preserved over time.
You might also add the Kuya.
Oh yeah.
Of the Palm Springs region who have classes, as well as the Serrano.
Oh yeah.
In San Bernardino County have classes. And
yeah.
Linguists who work with them.
Yeah.
Let me ask, I guess may not have caught it. Did you get samples from current populations to compare to the older ones?
Yeah, no, we didn't actually do that yet. We, a lot of the indigenous communities want to do that. So I think that's going to be for another study that we're for people who are, it's a bit difficult once you start doing that because you have to ensure ethically it becomes much more sensitive to make sure that you're, because an individual who exists in the present is going to represent the community even much more closely than individuals who are, say, 2,000 years ago. So we do want to do that because a lot of the indigenous tribal members want to do this, but we have to do it in a way that's very careful because it can inform so much on current relationships between different people and their very recent ancestors and people who are currently living today. It's more sensitive than even the ancient DNA.
But I'm sure you know through Dr. John Johnson that in the a decade or so ago, there were some studies like that done.
Yeah, we have a lot of mitochondrial DNA from a lot of people who were sequenced in the past. So we could track those people down, and actually some that we already know, to get autosomal DNA, so get their whole genome sequence, or at least genotype of throughout their rather than just mitochondria. Yeah, once you go beyond mitochondria, then it gets more ethically difficult, more sensitive, because mitochondria, you would just tell the person's haplotype, which can tell you something, but not as much as say the equivalent of 23andMe type genotyping on these people who are living today, because that can very directly tell you things about, your relatives and other sort of things. So, but yeah, definitely there are people have already been sequenced on a mitochondrial level. And yeah, getting the modern genotype data is something a lot of people are interested in, and how that relates to ancient people are very good questions. And Brian Hogan is actually doing a lot of this. He's has more recent ancient DNA. So these are people who are, say, 100 years old or 200 or 500 years old. And that will tell a lot more questions that a lot of the indigenous, a lot of the either Chumash tribal members or other tribal members are interested in, where you can start teasing together different stories, historical stories, and how this relates to oral history is more recent. So he has a lot of those studies about more recent history.
Also, I'm not sure if I caught what you said correctly, or but it seemed to suggest earlier on that you do have a DNA sequence that you associate with Uto-Aztecan speakers.
Yeah, so basically how this works in ancient DNA is that you are getting data from individuals who you presume spoke a particular language. And the problem is that if they don't have it written or other sort of things like that, then you're not certain. But we are assuming, in this study, we're assuming that people from Mexico, 1100 or 2500 years old, probably spoke Uto-Aztecan languages because they have very similar genetics to people who, one, they're in that region that was known to have spoken Uto-Aztecan, and two, they have very similar genetics to people who are in that region right after them, the Odom or Pima, who speak Uto-Aztecan today. And those, so we're using that as a marker for Uto-Aztecan related ancestry. So,
and then did you find people like that in Southern California?
Yeah, so we have, so the most recent individual who has the most amount of that ancestry would be someone who lives, there was an individual that that lived, I think, 500 years ago, who was near the Southern Channel Islands, kind of in the Tongva area today. And they have maybe 90% of that ancestry that is associated, similar to the kind of Mexico related ancestry.
Okay, so thank you.
No problem.
Here's a question for you. A couple of years ago, I sent a little tube of spit and a few dollars off to ancestry.com.
Yeah.
And they came back with, here's where I'm from, here's this percent of this and that percent of that. But then 2-3 years later, they sent me an update and they changed all my percentages.
Yeah, that's a good question.
Would this research have that same issue or change?
Yeah, so the thing with all of these ancestry companies is that they're using these algorithms that are based on people who live in the particular region. So I'll give a kind of a somewhat long answer to this, hopefully not too long. But a lot of the ways these companies do these is that they have people who live in, say, Ireland, in Italy, in Spain, in Southeast Asia, and China, etc. And they associate, Samoa, Tonga, Hawaii, etc. They associate your ancestry with that ancestry profile. So if they have a large number of people who are, say, Samoan or Polynesian, then they can kind of associate your ancestry with a Polynesian profile, or associate your ancestry with, say, British or Spanish ancestry. And the reason your ancestries change is because there are these clustering algorithms that get better over time as you have more and more data. So that's that leads to some of the changes in there. So with this study, definitely there are going to be some improvements over time as you get more and more samples of different ancient individuals who existed and sequence more closely. We get more and more refined hypotheses of what the actual ancestry is, what it looks like, and where it is spread out throughout the Americas. So this is very similar to what happened in our pictures of Europe and South Asia and other places, where we started off with a very small number of individuals that we sequenced, and as you get more and more, the picture becomes more and more clear. Then you have a clear ancestry type for a certain hunter-gatherer group in Eastern Europe or Western Europe and the differences between those. And you see the farming ancestry that becomes very clear. Then you see the steppe ancestry that becomes very clear and where that migrated into South Asia and into Europe. And as you get more and more individuals, you get more and more precise estimates of precise views of the ancestry, and you get a better and better historical hypotheses. So, so yeah, there definitely will be changes as we get more and more data, but this is kind of the current models. We're developing current models as we get more and more data.
Okay, thank you. Yeah, larger sample size has got to help.
Yeah.
Is there a group or an area that you'd like to get more information from specifically?
Yeah, very good question. So this, so these are very, very big archaeological questions that we're trying to answer. The same with kind of similar in Europe, where you're trying to figure out like who spread Indo-European languages. It's a very similar question to who spread Uto-Aztecan languages. I think the biggest places we would want to have data from are probably the Great Basin would be very helpful. We have some Great Basin individuals, but people from Colorado, Nevada, etc. A lot of that ancestry over time. Oh, sorry, I'll quit my speech area. Yeah, a lot of those individuals over time, and then probably Central California, just getting a better grasp of the history there would require a lot more individuals from around that region. So this study had a lot of interesting hypotheses and some data that's generated. But I think once you start getting more individuals from that region, then you can start getting better grasp of that. So I think those would be some of the biggest places, maybe a bit more from the Central California Valley to start evaluating better these hypotheses about where the Uto-Aztecan languages might have spread. So we can, the goal is kind of to eliminate certain models that are wrong and then provide evidence for different other models. So yeah, those would be some of the places that are really important, I think.
How's your data collected? Is it like Stephen said, you spit in a tube, have somebody, what do you collect to get this information?
Yeah, so usually you get, so those are, spitting in the tube would be people who exist today, but for these data are usually archaeologists are working with indigenous communities and getting bones from the area and skeletal material from the area. So what we do, what was done in this study is that we're working with an indigenous group. Either they are involved with the excavation of the skeletal material, and then they kind of bless it, and we sequence the material, and usually the bones are then repatriated and reburied. Sometimes they're in museums that were they were collected in the past, and then we try to connect them to the tribe now. So that's usually how it goes. There's some archaeology and indigenous groups that are working together and getting bones from these different areas. And if the tribe is amenable to it, then we'll study those bones as they come up.
Thank you.
No problem.
Are there any other questions? Okay, if not, Nathan, I'm going to be sending you this thank you certificate for tonight's
Oh, thanks.
talk. I'll be putting that in the mail to you. I appreciate
Yeah,
it.
that'd be great.
And I want to mention that the in September, I can find it right. Oh, here it is. We're going to be dark on June and July. July and August. We'll be back in September 14th. Dr. Philip DeBarros, the title of his talk is Exciting Pottery Discoveries in the Coachella Valley, 14th Century Ceramic Firing Pits in the City of Coachella. And that's the title of the talk for September. I hope everybody has a pleasant evening. I again thank Nathan. Have a safe drive home, everybody. And thank you all for joining us.
Thank you.
Thank you very much.
Thank you, Nathan. That was really good.
Thank you, Nathan, and thank you everyone for attending. And we look forward to seeing you all here in September.
Thank you.